Very good morning to students. So today we are going to start a new unit, that is drugs acting on the autonomic nervous system. Under this unit, the first topic is regarding the drugs acting on the cholinergic system. So in this session today we shall be discussing mainly about the transmission and receptors involved in the cholinergic system. So this topic we shall be covering under the following headings, that we shall have a brief introduction to autonomic nervous system, and we shall be mainly concentrating on the cholinergic transmission, cholinergic receptors, and the actions of acetylcholine. So first we shall look at a brief introduction to ANS. So you had already discussed a lot about the autonomic nervous system in your first year classes. So I'm not going into the discussion very elaborately. I'll be only discussing the points which are relevant for your understanding of the pharmacological aspects of the autonomic nervous system. So already everybody knows all these aspects which I'm going to discuss in the coming slides. That is, nervous system is divided into two parts, that is central nervous system and the peripheral nervous system. The peripheral nervous system is again subdivided into the somatic nervous system and the autonomic nervous system. Somatic nervous system, all of you know, they supply the skeletal muscles, and the neurotransmitter which is important here is the acetylcholine. On the other hand, the autonomic nervous system supplies all the other organs, and the neurotransmitter involved are acetylcholine or noradrenaline. The ANS parts also you are aware, that is the efferent nerve fibers, central connections, and efferent fibers. The motor efferent limb of the ANS is divided into some sympathetic nervous system and the parasympathetic nervous system. So you already know the features of sympathetic nervous system and the parasympathetic nervous system. So we shall just have a quick recap. Sympathetic nervous system or the adrenergic nervous system is mainly involved with the fight or the flight response. And the features associated with sympathetic nervous system are an increase in the blood pressure, increase in the heart rate, increased glucose and blood supply to skeletal muscles, mydriasis, bronchodilation. On the other hand, the parasympathetic nervous system or the cholinergic nervous system is mainly involved with the resting stage and with the assimilation of food. So it is mainly associated with decreased heart rate, decreased blood pressure, meiosis, insulin release, and glucose utilization, and bronchoconstriction. So it is opposite effects. Both these systems are having opposite effects. Now we are going to see the steps in neurohumoral transmission. So all of you know that the nerves transmit their messages across synapses and the neuroeffectors junctions by the release of humoral messengers. These humoral messengers are the chemical messengers. That is why it's called the neurohumoral transmission. The messages across the nerves, it is transmitted by the release of chemical messengers, which are called as the humoral messengers. That is why this transmission is called as the neurohumoral transmission. So we shall just briefly see what are the steps involved in the neurohumoral transmission. This also you have covered in your previous classes in the first year. So the first and the foremost step is the impulse conduction in the prejunctional neurons. So you can see in the figure, there is a prejunctional neuron. This is a postjunctional effector organ. So the first step is the impulse conduction in the prejunctional neuron. Already you have learned very elaborately how an action potential is formed in a neuron and so on. So we will not be discussing anything further on that. The first step is impulse conduction in the prejunctional neuron. The second step is the transmitter released from the neuron. You can see the transmitters being released. The red circles that you can see, the red spheres, these actually depict the transmitters. The transmitter released from the prejunctional nerve ending. These transmitters then combine with specific receptors which are present on the post-junctional effector organ. This post-junctional effector organ can be anything. It can be a nerve, it can be a muscle, it can be a blood vessel. Okay, so the transmitters that are released from the pre-junctional neuron, they combine with the receptors which are present on the post-junctional membrane. And then once the receptors combine with the transmitters combine with the receptors, an activity will occur, or the effect will be occurring at the post-junctional um, organ. So depending upon what kind of an organ it is, it can be different effects. So if it's a nerve, then there will be a transmission of a nerve impulse. If it's a muscle, it could be a contraction of the muscle. If it's a gland, it could be a secretion of the gland. So depending on the type of organ that it is, there will be different types of action. Now, once the receptor, the transmitter combines with the receptor and brings about an action in the post-junctional effector organ, the action of the transmitter has to be terminated. Now, how does the termination occur? The termination can occur either by the metabolism of the transmitter. That is, for example, in the case of acetylcholine, the termination of the action of acetylcholine occurs by the metabolism of the acetylcholine. Even in the case of noradrenaline, a part of it gets metabolized and then that action gets terminated. Or the action can be terminated by the active reuptake of this transmitter back into the prejunctional neuron. So you already know that they were released from the prejunctional neuron. Now when they're taken back into the prejunctional neuron, the action will be terminated. This is called as the reuptake into the prejunctional neuron. So noradrenaline is also taken up into the prejunctional neuron, and this also terminates the action of noradrenaline. So you can see the termination of action of noradrenaline is occurring by either metabolism as well as by the reuptake mechanism, whereas the termination of action of acetylcholine is occurring only by the metabolism. Now that is about the neurohumoral transmission. This is a figure that is showing. This is a prejunctional neuron. This is a postjunctional neuron. It is also a prejunctional neuron. And depending upon the transmitter that is released, you can see the transmitter being released. You can have different effects. So if it's the excitatory transmitter, you will be having a excitatory postsynaptic potential, and you will have excitation of the postsynaptic nerve. If it's an inhibitory neurotransmitter that is released from the prejunctional neuron, you will have an inhibitory postsynaptic potential, and you will have the inhibition of the postsynaptic postjunctional neuron. So this is a figure showing what I have just told you in the previous slide. Now let us have a glimpse of the co-transmission. So on stimulation, you have seen that the nerve is releasing a transmitter. So other than the primary transmitter, most of the nerves, they also release some other substances along with the primary transmitter. And these can be substances like the ATP, the vasoactive intestinal peptide, VIP, neuropeptide Y, NPY, substance P. These are called as the co-transmitters, and these co-transmitters also are released, and they bind with their corresponding receptors on the post-junctional effector organ and bring about their effect. So this is an example. You can see acetylcholine. It is the main, the primary neurotransmitter. It is released, gets bound to its receptors, and along with that you can see the release of a co-transmitter, that is VIP, vasoactive intestinal peptide, which is released along with it, and it also goes and binds to its corresponding receptors on the post-synaptic effector organ. So this is the co-transmission, and co-transmission occurs in many of the neurons. Now that is about the brief introduction of adrenergic nervous system. Now we shall proceed on to the topic proper, that is cholinergic system. Otherwise it is called as the parasympathetic nervous system. So let us look at the main neurotransmitter. The neurotransmitter of cholinergic system is acetylcholine. Now we shall see where are the sites where the cholinergic transmission occurs. So you have to learn these sites by heart. So first, all preganglionic sites, whether it is parasympathetic or sympathetic, preganglionic sites. All the postganglionic parasympathetic sites. Postganglionic sympathetic supply to sweat glands and some blood vessels. Ganglia. either sympathetic as well as the parasympathetic. So in the ganglia, you mainly have the cholinergic transmission, even in the adrenal medulla. Then in the skeletal muscles, in the CNS, different parts like cortex, basal ganglia, spinal cord are the sites of cholinergic transmission. So you have to learn these sites by heart. Now we can look into the steps of cholinergic transmission. So the first step is a synthesis of the neurotransmitter. I told you the neurotransmitter involved in cholinergic transmission is acetylcholine. So first step is a synthesis of acetylcholine. So how is it synthesized? So it is synthesized by the following steps. The choline is the active raw material which is required for synthesis of the acetylcholine. So the choline is actively taken up into the neurons by a sodium choline cotransporter, and it is acetylated with the help of ATP and coenzyme A in the presence of the enzyme choline acetyl transferase into acetylcholine and coenzyme A. So you can see first step is acetyl coenzyme A, which is formed from ATP, acetate and coenzyme A by the acetate activating reaction. The choline, which is taken up by the sodium choline cotransporter into the neurons, combines with acetylcholine, acetyl coenzyme A in the presence of the enzyme choline acetyl transferase to result in the synthesis of acetylcholine and coenzyme A. So the synthesis of the neurotransmitter has occurred. Now we go on to the next step, that is the storage of acetylcholine. Once it is synthesized, acetylcholine enters the synaptic vesicles. You can see these are the synaptic vesicles which are present in the nerve terminal, and the entry of the acetylcholine into these vesicles occurs with the help of a transporter which is called as the vesicle associated transporter or VAT. Once it is entering the vesicles, it is stored there. You can see synthesis is occurring, and after synthesis it is entering the vesicles and it is getting stored. Next step is the release of acetylcholine. So acetylcholine release occurs in small quantity from the vesicles by the process called exocytosis, and this occurs in response to a nerve action potential. So whenever there is an action potential reaching the nerve, then small quantities of acetylcholine will be released. Once it is released, acetylcholine acts on the cholinergic receptors. The cholinergic receptors we will be learning in detail in the following slides. So it is binding to the cholinergic receptors. I have depicted the cholinergic receptors by N and M. You will see what they are later on in the slides. They bind to these receptors and bring about the action of acetylcholine on the postsynaptic effector organ. Now, the next step is destruction or termination of action of acetylcholine. So I told you in the previous slides, the termination of action of acetylcholine occurs by metabolism. So it is rapidly metabolized by an enzyme which is called as the acetylcholine esterase. So just see how it is abbreviated ACHE. You have to be careful with this acetylcholine esterase. This is the short form of the enzyme. It is metabolizing acetylcholine into choline, which is again recycled for synthesis of acetylcholine, and the other part is acetate. Now we have to see there are two types of acetylcholine esterase. One is called as the true choline esterase, and the other one is called as the butyryl choline esterase or the pseudo choline esterase. So we shall see what is the difference between these types of choline esterase enzymes. So you can see here the distribution. So the true cholinesterase is present in all the cholinergic sites in RBCs and gray matter. On the other hand, the pseudocholinesterase is present in plasma, in the liver, intestine, and in the white matter. The basic difference between these the two enzymes is that acetylcholine is very fast hydrolyzed by the true cholinesterase, whereas the hydrolysis of acetylcholine is very slow by the pseudocholinesterase. On the other hand, you can see that methacholine is hydrolyzed very slowly by the true cholinesterase and not at all hydrolyzed by the butyryl cholinesterase. One basic difference here now is that there are some ingested esters. You can see here this benzoyl choline and butyl choline. These are some of the esters which can be provided into the body from outside. These are not at all hydrolyzed by acetylcholine esterase, whereas they are hydrolyzed by the pseudocholinesterase. These are the basic differences between the true cholinesterase and the pseudocholinesterases. Now, how is it inhibited? This enzyme, that is cholinesterase, is inhibited by a drug that is called as physostigmine, and the pseudocholinesterase is inhibited by organophosphates. Now, the main function of cholinesterase is mainly termination of acetylcholine action. You have seen that it hydrolyzes the acetylcholine very fast. So that is the main action. Whereas the action of pseudocholinesterase is hydrolysis of ingested esters like benzoyl choline and butyl choline. So with that, we have completed the cholinergic transmission. Now we shall see some of the modulators of cholinergic transmission. Modulators means the drugs which are either inhibiting or increasing the cholinergic transmission. So there is a drug that is called as hemicholine. It blocks the choline uptake and depletes the acetylcholine. I told you the first step was choline uptake. So this is being inhibited by hemicholine. Another drug is vesamicol, which inhibits the uptake of the acetylcholine into the storage vesicle. That is mainly blocking the VAT transporter. So you can see vesamicol, it is having an inhibitory action on the VAT transporter. Then you can see the botulinum toxin. This toxin inhibits the acetylcholine release from the nerve terminal. So you can see botulinum toxin is having an inhibitory effect on the release of acetylcholine. On the other hand, there's another toxin called as the black widow spider toxin. It causes massive release of acetylcholine. But because of the massive release, finally there's a depletion of acetylcholine. Yeah, these are the drugs which you have to learn. You have to learn to draw the figure and to see, and you have to learn how to mark these drugs, where these drugs act on the steps of cholinergic transmission. So at this point, I like to mention something about botulinum toxin also. So this toxin is of two types, that is A type, that is botulinum toxin A, and botulinum toxin B. It is mainly produced by the Clostridium botulinum and is associated with the botulism, that is a kind of food poisoning. However, there are some therapeutic uses also of botulinum toxin A. But the one thing that has to be put into mind is that very localized injections of very minute amounts. It is available in the brand name Botox, and that is used for the therapeutic purpose. So you have to be very careful that it should be very localized injection as well as for in very minute amounts. Now, for what conditions it is used? It is used mainly for disorders like blepharospasm, spastic cerebral palsy, strabismus, spasmodic torticollis, nystagmus, and axillary hyperhidrosis. So, in this case, it's an excessive cholinergic activity. So all these conditions, in all these conditions, it's an excessive cholinergic activity. So I already told you botulinum toxin, it is an inhibitory, it inhibits the cholinergic acetylcholine release. So it can be used in all these conditions in very localized injection and in very minute amounts. If the injection is inappropriately given, or if large amounts are given, then it can lead to side effects or adverse effects like ptosis, diplopia, facial swelling, dry mouth, and all. And one more very important or very popularly used situation of Botox is, all of you know, it is to relieve the age-related wrinkles, especially in the movie stars and all people in the show business. You can have seen that they are using this Botox for removing the age-related wrinkles. I have put up a picture here. This is the age-related wrinkles, and after the administration of Botox, you can see that the wrinkles have been eliminated. Now we'll go to the next part, that is the cholinergic receptors. So cholinergic receptors are basically of two types: muscarinic receptors or the M receptors and nicotinic receptors or the N receptors. All the muscarinic receptors are a type of G protein coupled receptors, whereas the nicotinic receptors are ligand gated cation channel receptors. Okay, so already you have learned in detail about the GPCR and the ligand-gated receptors, how they work, function, and all. So you can see this is a structure of the GPCR, and this is an example is muscarinic receptors. Nicotinic receptors, on the other hand, they are pentameric rosette-shaped structures, and they have a cation channel in the center. So whenever this receptor is stimulated, this cation channel will be opening and the ion flow will be occurring and depolarization will occur. This is how, this is the detail I think you have already studied in the chapter on receptors. So first we shall look at the muscarinic receptors. Muscarinic receptors are again divided into 5 subtypes: M1, M2, M3, M4 and M5. So you can see I have made this bolded the M1, M2 and M3. This is because these are the main 3 subtypes. M4 and M5 are less significant. They are present in neurons of the brain, but we'll be principally concentrating on the first 3 receptors. It is M1, M2 and M3, because most of the actions of the cholinergic system are being conducted through these 3 receptors. These receptors are stimulated by muscarine, which is the agonist on all these receptors. They are blocked by atropine. The atropine is the antagonist on all the muscarinic receptors. So each subtype, I told you each receptor is a type of G protein coupled receptor. And when you were studying the G protein coupled receptor, you have studied the function through different effector pathways. It can be cyclic AMP pathway or IP3DAG pathway or through the channel regulation. So here in this case, M1 and M3, it functions mainly through the IP3DAG pathway, whereas the M2, it is mainly functioning through the channel opening or channel regulation. So you have to know only this much at the undergraduate level, that they are all G protein coupled receptors and they function through either the IP3DAG pathway or through the channel regulation. Now we shall look into each subtype and with respect to their location, function, selective agonist and selective antagonist. This table is actually the crux of this chapter. So I'll tell all of you, it's an advice to all of you that you can make this chart and stick it somewhere in your room near your study table. So that you can always see this every time you see this, you can always keep on revising. So this is actually the crux. So to understand the cholinergic drugs also, you will require this table and the next table on the nicotinic receptors also. So this is the very most important part so far discussed. So M1. So M1 is present on the autonomic ganglia, on the gastric glands, intestine and the CNS. In the autonomic ganglia, they cause depolarization. In the gastric glands, they cause histamine and acid secretion. In the intestine, they cause intestinal secretion. Whereas the action on the intestinal secretion is minimal, but the action is to increase the secretion. In the CNS, they cause a varied actions like relating to learning, memory, motor, and functions. This is about the M1. The selective agonist is oxotremorine and the selective antagonist is pirenzepine and dilenzepine. I told you the agonist on all the receptors is muscarinic and the antagonist on all the receptors, muscarinic receptors, is atropine. This oxotremorine is selective for M1. The selective agonist on M1, and similarly pirenzepine and dilenzepine are selective antagonists on M1. They don't act on the other M2 or the M3. They are acting only on the M1 receptor. The M2 receptor, again, it's a very important receptor. You can see it is present on the heart in different parts, SA node, AV node, atria, and on the ventricles. It is also present on the cholinergic pre-junctional nerve endings. So I told you the nerve ending from which the transmitter is released. It's a pre-junctional nerve ending. So the cholinergic pre-junctional nerve ending also, it is present. I can show you here. We had a picture here. See, this is the pre-junctional nerve ending, and this is the muscarinic M2 receptor, which is present on the pre-junctional nerve ending. So you can see other receptors, they are present on the post-junctional effector organs, but this M2 receptor is present on the pre-junctional nerve ending. So that is one of the sites of locations. It is also present in the CNS and in some cases in the visceral smooth muscles also. So the heart, it causes decreased rate of impulse formation, conduction velocity and contractility of the heart. So all of it is decreased. In the cholinergic nerve endings, it decreases the acetylcholine release. So these are called as the autoreceptors because they are present on the same type of nerve terminal from which they are released. And their function is to inhibit the further release of the acetylcholine. In the CNS, they are associated with mainly tremor and analgesia. Visceral smooth muscles, they cause contraction. Selective agonist is methacholine and selective antagonist on M2 receptors is methotrexate and tryptophan. Now let us move on to the M3 receptors. They are mainly present on the visceral smooth muscles. So you can see that visceral smooth muscles are having both M2 as well as the M3 receptors. It is also present on the iris, on the ciliary muscle, on the exocrine glands and the vascular endothelium. So concentrate more on this exocrine glands, vascular endothelium. So you can see here contraction is caused of the visceral smooth muscle. So visceral smooth muscle can be contracted either by the M3 receptor action or by the M2 receptor action. So in different organs, the contraction can lead to different effects. So for example, bronchial constriction, it can lead to bronchoconstriction. Similarly, the gut contraction, it can lead to the evacuation of the bowel. It can lead to increased peristalsis and evacuation of bowel. Similarly, ureter peristalsis can be increased and because of the detrusor muscle contraction, evacuation of the bladder can occur. So this is what a contraction of the visceral smooth muscles is leading to. Now the contraction of the circular muscles of the iris, it leads to meiosis or the pupillary constriction. Ciliary muscle contraction leads to spasm of accommodation. So you already know that the ciliary muscle is the one that is responsible for the accommodation of the eye, and it has to relax and contract to have a proper accommodation. But if it is in a contracted state, you will have spasm of accommodation. Okay, then there is an increased aqueous outflow and fall in IOT. The contraction of the ciliary muscle is also associated with an increased outflow of the aqueous humor and subsequent fall in the intraocular tension. So the cholinergic transmission is actually better for a patient with glaucoma because it increases the outflow and decreases the intraocular tension. On the exocrine glands, it increases the secretion of these glands. Vascular endothelium. So it causes the release of nitric oxide from the vascular endothelium. It is called as the endothelium dependent relaxing factor because it causes the vasodilatation of the blood vessels. So the endothelium derived relaxing factor or the nitrous oxide, it causes the relaxation of the blood vessels and leads to vasodilatation. So you have to see here one thing, that visceral smooth muscles, it is present in the smooth muscles of the blood vessels also. The contraction causes vasoconstriction, but here the vasodilatation is caused mainly because of the action of the EDRF, which is present, which is released from the vascular endothelium. And this is the principal action on the blood vessel. That is the action that is mediated through the EDRF, is the principal action of the cholinergic system on the blood vessel. That is mainly it causes a vasodilatation. Selective agonist beta-nicol and selective antagonist on the M3 receptor is solifenacin and darifenacin. So this you will learn by heart. Now let us move on to the nicotinic receptors. They are of two subtypes, NM and NN type, and they are all stimulated by nicotine. That's an agonist, and as I already told you, they are all ligand-gated cation channels. Now we'll see the difference between the NM receptor and the NN receptor. Before that, just remember that acetylcholine, that is a neurotransmitter of the cholinergic system, it will act on the nicotinic receptors only at high concentration. That is one important thing that you have to remember. Only at high concentration it will act on the nicotinic receptors. Otherwise, it will be having the muscarinic actions only. So the NM receptor is present on the neuromuscular junction or the skeletal muscle end plate, and it causes the contraction of the skeletal muscles. So always remember, Acetylcholine causes the contraction of smooth muscles by its actions on M2 or M3 receptors, whereas it causes the contraction of skeletal muscles by the action on NM receptors. This is a very basic mistake that most students do. So this is one important point. I want all of you to remember this always till the end of this topic, as well as the till the end of your syllabus. You have to remember this basic difference. NM is present on the skeletal muscle, and the contraction by acetylcholine on the of the skeletal muscle is caused by its action on the NM receptor, whereas the smooth muscle is contracted through M2 or the M3 receptors. Now, the selective agonist is PTMA and selective antagonist is D-tubocurarine, that is skeletal muscle relaxant, and a toxin that is alpha bungarotoxin. NN, again, it is acting on, located in the autonomic ganglia, that is both sympathetic as well as parasympathetic ganglia. It is located adrenal medulla and in CNS. So, the stimulation causes depolarization and release of catecholamine. Okay, that means catecholamine means noradrenaline, adrenaline and all, and this can cause a high rise in the heart rate and BP. Okay, and the selective agonist is DMPP and the selective antagonist is hexamethonium and trimethophan. So at this point, just remember that nicotinic receptors are stimulated at high concentrations of acetylcholine. Now, with this background, that is the actions and the locations of muscarinic and the nicotinic receptors, we will move on to the actions of acetylcholine. Acetylcholine is a neurotransmitter of the cholinergic system, so its actions will be a sum of both muscarinic and the nicotinic action. So far, whatever actions we have discussed under the table, the two tables, which will all be depicted here once again. So we'll just have a look at it. So first we'll look at the muscarinic actions. So the muscarinic actions on the heart. So I told you on the heart, you have got the M2 receptors on the SA node, AV node, and mainly on the atria. So you can see that it causes a decrease in the heart rate. By acting on the M2 receptors on the SA node, it causes a decreased AV conduction velocity, and it also causes a decrease in the atrial contractility. Now, ventricular contractility is also decreased, but the effect is not as much marked as the effect on the atrial contractility. One more thing is that because this vagal fibers, they are very non-uniformly distributed in the atrial fibers. Because of this, what can happen is that it can lead to an atrial fibrillation. That is, parts of it will be contracting fast and the parts of it will be decontracting, the contracting less. So because of this inhomogeneity of the distribution of the vagal fibers can lead to atrial fibrillation. So this concept is a little bit difficult and you need not bother about it at this point of time. Just remember that it can cause atrial fibrillation because of inhomogeneity of the distribution of the vagal fibers in the atria. Now the blood vessels. So I told you the principal actions on the blood vessels is mainly because of the endothelium derived relaxing factor that is released from the vascular endothelium. So this is causing a vasodilatation of the blood vessels, and this will lead to a decrease in the blood pressure. Now, if the vascular endothelium is diseased or it is damaged because of some disease, then this relaxing factor will not be released. So what will happen? Now only vasoconstriction will occur because of the action of the vascular smooth muscle contraction. Okay, so the principal action is mainly through the vascular endothelium, which causes vasodilatation. Only if it is damaged, then only the action of the vascular smooth muscle contraction and vasoconstriction will come into picture. Okay, now the smooth muscle. The smooth muscle contraction can occur mainly because of its action on M2 or the M3 receptors. I mentioned only M3 here, but there's also M2 action also, and all of it is contraction only. And I told you various example: bronchoconstriction, abdominal cramps, and evacuation of the bowel, then increased peristalsis of the ureter and the evacuation of the avoiding of the urine. All this is a result of the contraction of the different smooth muscles. On the glands, it's a secretion of the exocrine glands and can lead to sweating, salivation, lacrimation, increased tracheobronchial secretions, and so on. Intestinal secretions are not that much affected, and milk and bile secretions are not at all affected by the cholinergic transmission. On the eye, already I told you it causes meiosis, and it can cause a spasm of accommodation, increased aqueous outflow, and reduction in IOT because of the contraction of the ciliary muscle. This is just a summary of what we have told in the previous slides. Now let us look at the nicotinic actions. So already we know NM receptor, by its action on the NM receptor, acetylcholine can cause a contraction of the skeletal muscle. Then by its action at very high doses, it can cause an action of the NN receptor present in the autonomic ganglia, and this can lead to the release of catecholamines, and this can lead to increase in the heart rate and the blood pressure. So at this point, you have to remember that at low doses or at normal doses, the acetylcholine acts only on the muscarinic receptors, and there by acting on the M2 receptor, it causes a fall in the heart rate, and by acting on the M3 receptors on the vascular endothelium, it causes a fall in the blood pressure. Now, when high doses are given, and after you give high doses of acetylcholine and you block all the muscarinic receptors by atropine, then what will happen? Only the nicotinic receptors will be stimulated, and in this case what will happen? You will have more release of catecholamines, and this will result in tachycardia and rise in BP. So this is one important point that you have to remember. High doses of acetylcholine given after blocking muscarinic receptors with atropine will lead to tachycardia and rise in BP. Otherwise, at low doses, acetylcholine will cause a fall in BP as well as in a bradycardia. Okay, so I hope that is clear to all of you. So these are the important questions. Steps of cholinergic transmission, you have to draw the diagram and then you have to explain. You have to also know to mark the different sites of action of the different modulators which I have mentioned. Hemicolinium, besamicol, the two toxins that I mentioned, botulinum toxin and the black widow spider toxin. You should know how to mark the actions on the diagram. And the muscarinic receptors, you can learn in the tabular column form, location, function, selective agonist and selective antagonist. Similarly, you have to learn about the nicotinic receptors. And I would advise you to make the two charts of these receptors and stick it in your room so that you can always revise it and see it again and again. And then actions of acetylcholine you have to write under muscarinic actions and nicotinic actions. So I hope you have understood this session, and if you have any doubts you can just feel free to contact me. Okay, thank you all for your patient hearing.